So hi and welcome everybody to this introduction for cell signal transduction. And today we're gonna talk about cell signaling. Here's some literature if you're interested. And let's dive right into it. So what exactly is signal transduction? Well, our cells use either electrical, chemical or regulatory mechanisms to transduce information through the organism, which concludes to a response on a cellular level. The forms of these signals can either be chemical, electric or mechanic. Why do we need cell signal transduction? Well, our body needs to adapt to environmental and external changes. And our body has to coordinate our cell functions, for example, the cell number, morphology, location, or function. And we make a difference between two different cell signaling ways. One is the intersignaling, which means that one cell is communicating with another cell, which leads to an intrasignaling. This means signaling paths that are introduced in the cell itself. Intercellular signals, which means cell to cell, can be either electrical stimulation, for example via gap junctions, or they can be chemical via hormones, growth factors or neurotransmitter, which bind to a receptor. And then again, the intracellular signals, there we have different components in the cell. First of all, we have the stimuli, then we have receptors, we have transducers, amplifiers, messengers, sensors and effectors, which all together lead to a cellular response. And altogether, they are called signalosome, all those toolkits here. And the signalosome is a toolkit which is characterized by a high plasticity to respond properly on changes. The signalosome is highly cell-specific. This means that the same stimuli can lead to different cell responses depending on the type of cell receiving the signal. Let's talk a little bit in more detail about our different components. First of all, we have the stimuli, and a cell can release stimuli via diffusion, vesicle or exocytosis. The stimuli can then act autocrine, juxtacrine, paracrine or even endocrine if it's released into our blood system. We have different receptors on the surface of our cell and inside the cell itself. The reason behind this is that there are either hydrophilic stimuli, which cannot pass our cell membrane, and therefore we need receptors on top of our cell, and we have lipophilic stimuli, which can pass the membrane, and that's why we have also receptors inside of our cell or inside our nucleus. There are different kinds of receptors on the cell surface. Receptors, we have ion channels, single membrane spanning receptors and multiple membrane spanning receptors. And on the inside of our cell we have mostly hormone receptors, because hormones can normally pass our cell membrane because they are lipophilic. Then we have transducers. What is a transducer? Well, when there's a signal from outside coming, there's a problem, excuse me, because the stimuli from outside the cell speak a different language than the signals provided inside of the cell. And transducers are translating the outside signal to the internal language of the cell. For example, we have this inactive G protein, which is activated by the stimuli from outside and then passes the signal inside the cell. Amplifiers like this G protein here are simply enhancing the signal inside the cell. They do so by producing diffusible intracellular molecules that transport the information from the cell surface to the internal sensors, and those molecules are called messengers. And then again we have sensors which can detect those messengers and use the information to stimulate effectors, which then start different cellular processes. Like, for example, they can start the transcription and translation of the DNA. We have different regulation points here. We can regulate our enzymes, for example, by the amount of enzymes available or the activity of enzymes. And then again, we can modify our enzymes via glycosylation, phosphorylation, oxidation, and many more. And on gene expression levels, we can regulate the transcription, for example, via DNA methylation, or we can regulate our translation, for example, with the help of our ribosomal activity. Whenever there is a signal induced, there comes a time when the cell is no longer in need for the signal, so we somehow have to turn it off again. And how can we do this? We have different attack points here. First of all, we have our receptors. We can reduce the amount of receptors available on the cell, for example, via sequestration or via lysosomes. And then again, we can inactivate our receptor with an inhibitor, or we can inhibit signal proteins. In our body, most signals are transduced via phosphorylation through kinases, and if we want to turn this down, we can use phosphatases, the counterpart of kinases, which dephosphorylate our proteins again, turning them inactive. And last but not least, we can reduce or decompose our messenger molecules to stop the signal cascade. I hope this was somehow informational for you, and I hope to see you again. Have a wonderful day.